All right, everyone. Thank you for being here. Our next speaker is going to be Dr. Carl Kearns. Dr. Kearns is an assistant professor in the animal science department at Iowa State University, and he is going to talk to us about what's new in boar research.
Okay. Thank you for that introduction. So today we'll be talking about the present and the future of boar fertility testing. Kind of talk about where we are today in examining boar potential fertility, and then getting into where we're going in the future perhaps, and being able to tell at the boar stud which boar has higher or lower fertility, and then the implications that we can do with that. Okay. So give a little bit of background about boar fertility, talk about the pathway to fertilization success, and then we'll talk about the current fertility diagnostics, and then get into the currently available technologies and the not-so-distant future. So ultimately what drives the research question in my lab is what predestines the fertilizing spermatozoon. If we think about a sow, we're inseminating with over 2 billion sperm cells typically, and what makes of those 2 billion sperm cells 20 to 40 sperm cells, the lucky ones to go on and form new life. And so there's certain characteristics that might be present in some sperm cells that make them maybe a little bit more competitively advantaged than other cells that don't make it later on. So we know that boar fertility plays an important limiting role in the reproductive performance of swine operations. Dr. Billy Flowers did a really nice summary of this, not only on the pig side, but also with beef cattle and dairy cattle and sheep, showing that we've done a really good job of creating a female that has a little bit better confined fertility. And so we see that we have a uniform female compared to the male side, and we see that the male has a wide variability both in pregnancy rate as well as the total number born live. And so if we think about it, if we're using one AI boar across 300 to 800 sows, we can have a large impact if we choose a boar that has lower fertility compared to other boars in his cohort. And we'll say, well, we use pooled semen, and that helps us get away from that situation. But sometimes we know that there might be situations where lower boars can actually lower the fertility of a pool. And so the Mashoffs and Acuity Genetics did a really good job summarizing this with current genetics, fast forwarding 10 years later from what Dr. Billy Flowers would have shown, and showing that even after standard motility and morphology characteristics are met, they're in the gray and black lines, over 80% motility and morphology, that we still have about 25% of boars that have less than 80% pregnancy rates. And so that means we have quite a bit of low-hanging fruit to go after to try to boost the overall herd performance. And we know that this is true not only on the conception rate side, but also present on the total number born. We see quite a bit of variability in the total number born from the sire side. And so this all comes together to show that there's different quality metrics that we can look at. And oftentimes one of the first things we look at when a semen collection comes into the lab is motility. And so oftentimes I refer to motility as in the workplace. We look at motility and look at how fast are they swimming straight and whatnot. And we say that just because they have motility that they're fertile. But that could be anything further from the truth. If you show up to the workplace and watch people walk from their car and into the building that they're working in, and assuming that shorter time period is how good they'll do their job for the rest of the day, that's not fair to humans, and it's also not fair to the sperm cell. And so we have to make sure that they can show up and do the job necessarily, but then can they do molecular events after that to be successful in fertilization. And so if we think about the pathway to fertilization success, one of the first things that hopefully the sperm are going to after insemination is the oviductal sperm reservoir. So this oviductal sperm reservoir, they get there in about 10 to 15 minutes, and they bind to glycans or sugar molecules there in the oviduct and just hang out there until the time of ovulation. We want to make sure that we have sperm present before the time of ovulation so we can maximize the number of oocytes or eggs that are later on fertilized. After they're released from the oviduct, hopefully they go through a process called sperm capacitation. Sperm capacitation was first defined in 1951 by Austin and Chang, both independent of each other. And there's many attributes and pathways involved with sperm capacitation, but the main things that we want to keep in mind here is that there's ions that are coming in, calcium ions that are coming in, bicarbonate that's coming in as well, and then ultimately allowing for detection of the oocyte after a series of events. And so they first defined capacitation as the acquisition of the capacity to fertilize an oocyte. And so that in its very essence has a functional characteristic to it. Do they have the capacity or incapacity to fertilize an oocyte? And oftentimes in the decades since the first discovery of sperm capacitation, we've put a very biochemical definition to capacitation. We've had multiple different various attributes that we think makes a capacitated sperm cell a capacitated sperm cell. And then when we do knockout studies, we've seen that actually those attributes aren't needed, and that such as protein tyrosine phosphorylation. Protein tyrosine phosphorylation used to be believed as the hallmark, the ultimate point of sperm capacitation. Visconti and his team that discovered that back in the 80s have since done knockout studies, knocking out the gene for that event to happen. And they've seen that they actually don't even need protein tyrosine phosphorylation to have fertility. And so it's really making us question what is sperm capacitation, what is involved in sperm capacitation, and I'll be presenting a little bit of that on the way today. But sperm capacitation is on the opposite ends of a continuous spectrum, that sperm capacitation ultimately leads to cell death. And so the sperm cells on this careful balance of making sure they don't create too much reactive oxygen species, that those reactive oxygen species actually help it progress through capacitation, but at the same time they can die from that. And so after a sperm cell actually goes through sperm capacitation, the lifespan of that cell is quite limited in the matter of seconds to minutes. And after we go through sperm capacitation, hopefully we've made it to the oocyte and can actually make it through the outside of that oocyte, the zona pellucida. And so even though we might have penetration of the zona pellucida, we have to make sure that the activating factors on the sperm cell can actually activate the oocyte and say, we're here, let's continue forward with embryogenesis. And there's quite a bit of studies showing that when these activation factors aren't very high, if there's low amounts of these proteins present, that the embryo cannot proceed forward into embryogenesis. So this all comes together to show that there's multiple dogmas that's been around for the last 2 centuries, and the two prevailing dogmas is illustrated here. Dogma 1 shows that every live sperm cell is fertile, and so if it's moving, if it has good morphology, that those are the fertile cohort. And the other dogma says that no, maybe there's only a small subpopulation that's actually capable of going through all the events necessary for fertilization. And so I give you a little bit of background on sperm capacitation and making it the way through the zona pellucida and oocyte activation to say when your car starts, there's multiple things that can go wrong when you try to start your car. If not everything lines up to actually allow you to start the car, same thing can happen on the sperm side. If you have one little piece out, then you cannot proceed forward with positive fertility. And there in subpanel C there, we see how we can identify that fertile cohort. So it's really captured by the two dogmas, in fact. We can look at motility and morphology, which are prerequisites. We have to have good motility. We have to have good morphology. But then the next level after that is biomarkers. What can biomarkers tell us about the cellular health that allows that sperm cell to go through sperm capacitation? And so this is an example of what some of those biomarkers look like. And so off to the right there, or to your left there, you can see what conventional semen analysis might look like underneath the microscope. And at first glance, it looks like most of those sperm cells are quite good, but then once you put the biomarker on there, you can start to see, oh, there's a sperm cell off to the right there that has a bent tail. And if you're just looking underneath the microscope, it can be really hard to see that off to the left-hand side there. There's other biomarkers that are present that show negative, bad sperm cells that might not necessarily have any morphology issues, but they are indeed sperm cells that are going to be recycled by the body. And they're shown in red, the ubiquitin tagging there. And so those sperm cells have been tagged by the boar, saying that there's something wrong with the sperm cell and that it needs to be recycled by the body. And so these sperm cells likewise are infertile and not capable of fertilizing an oocyte. And so there's been biomarker studies done to show that in high versus low biomarker instances that we have total number born differences, and as well as farrowing rates are affected as well. And so this kind of brings us into the zinc signature story, and we'll talk about this a little bit more in detail, but this is what my lab's been focusing on. And so we've shown that zinc is a key regulator of fertilization competency, and so we have to have zinc present to keep the sperm cells in a state that allows them to fertilize later on, but then we have to have them go through a series of events that actually allows them to release the ions so they can go through the last steps of fertilization. So getting into the currently available technologies. There's 4 major currently available technologies that we'll talk about. The first one is computer-assisted semen analysis systems, and so shorthand with that is CASA, and that allows us to look at the motility and the morphology. Then we get into flow cytometry that lets us look at the biomarker cell health and what attributes the cell might have that makes it good or bad. Then image-based flow cytometry kind of combines traditional microscopy along with the biomarkers so that we can see what's the localization of these various biomarkers. And then finally, pipetting robots. So this is an example of a CASA, and we'll have some metrics that we show on the next slide. But really a CASA comes down to having a microscope that's outfitted with a computer that can analyze individual sperm cells and look at how fast are the sperm cells moving. Are they moving in a straight or curvilinear position? And then really gives us a good idea of how many of these sperm cells are moving fast, progressively, or maybe a little bit slower as well. And a lot of these CASAs now can look at morphology as well. And so you get both motility and morphology, looking at the entire sperm cell in a very quick fashion. And so this is an example of some of those CASA outputs that you see. And so there's all kinds of metrics that come out of these that really let you hone into the individual sperm cell at the sperm cell level. And there's a lot of work that's being done in this field that particularly you can maybe look at cohorts of sperm cells. Right now we look at averages of what's the entire sample look like at the average level. But there might be some underlying features that we can dig out at the single cell level of saying that there's a cohort of sperm cells on this side that have a little bit different attributes than the other cohort. And so Minitubes came out with what they call the eFlow system, and this is a good system to basically have really standardized CASA analyzing abilities. One of the issues with chamber slides is that you can kind of tweak where you're looking at in that chamber, and you can kind of skew the results of an individual sample if you wanted. And so the beauty of the eFlow system is that you have a consistent system that is consistently looking at the same area. You have no user error in looking at a portion of the chambers that's off to the side, and as well as integrating the auto morphology features into it as well. And then it just came out with a portable solutions that perhaps in the sow barns or in smaller bore studs you can use what's called the androscope. And so this is a more economical solution if you don't have the number of bores to support a larger CASA system. And then getting into flow cytometry. So flow cytometry back in the day used to take an engineering degree to actually run to run the flow cytometer. Took up an entire room, as you can see here, and was really intricate to run. And so you can imagine that a cell biologist or andrologist can't really gain the tools and techniques to run something like this on an average day. And today there's small benchtop flow cytometers that you can quite easily run, have quite a bit of automation into them that allows us to look at samples on a day-to-day basis. And then building on that is the use of image-based flow cytometry. And so image-based flow cytometry uses the same mechanisms as traditional flow cytometry, and using the fluidics that lets you look at thousands of cells per second or per minute. And then ultimately that captures an image with each single one so that you can look at where the biomarker health status of it, is located on the sperm cell. And so when we talk about image-based flow cytometry, we also talk about the gating strategy used. So we want to make sure that we look at sperm cells that are in focus as opposed to out of focus, and then looking at single sperm cells per picture as opposed to multiple sperm cells. And then finally, making sure that the cells are laterally or anterior or posteriorly aligned as opposed to laterally aligned with the camera. And then building on top of that, we want to make sure that we're looking at a particular part of the sperm cell. And so how it came out with the zinc signature story that I'll get into here in a second is that we couldn't really tell the subpopulations until we looked at a masking strategy that let us look at one particular portion of the sperm cell. And so what we did with that is we created a masking strategy that let us look at the tail portion only, and that let us segregate out the sperm cells that we actually gate off, actually populate how many sperm cells were present in the 4 different zinc signature patterns. And so this is kind of an example of what we're able to do with image-based flow cytometry. Before, without image-based flow cytometry and the gating mechanism, that's off to the left-hand side what the histogram would look like. And we can't really pick up on very small, minute changes that are occurring. But then after we have the implementation of image-based flow cytometry, we can pick up on really small events and changes that are occurring. And so what we saw with the zinc signature assay is that the zinc signature is indicative of the time span left in that sperm cell to actually live. It tells the time point that they're at in sperm capacitation, and we saw 4 different patterns with that. We saw zinc, what we called zinc signature 1 had zinc localized to the entire sperm cell. Zinc signature 2 had just the middle portion of the tail and the sperm head, and then signature 3 with the mid-piece region only, and signature 4 with no zinc localization at all. And what we saw is that as these sperm cells go through a process that we call in vitro capacitation, where we're trying to mimic the same events that are occurring in the sow or gilt, that we saw them progress through these various zinc signature patterns. And we also saw localization patterns in the acrosome changing as well. And we can see how the acrosome was modifying throughout the capacitation, which is a traditional indicator of sperm capacitation. What's really cool to see underneath the microscope is the transition from a signature 2 to a signature 3 state. And so this occurs in the matter of 2 to 3 seconds. It's really hard to actually get video footage of this, because oftentimes when we look at fluorescence, it takes maybe 1 to 2 seconds just to capture one fluorescent image. And so if we want to capture zinc, another biomarker that tells us the plasma membrane integrity, and then the Herx DNA stain, it takes anywhere from 2 to 5 seconds just to get one time point there. And so you can imagine if you have a video footage that you're trying to get 30 images per second or even 60 images per second, we can't possibly get this with the epifluorescence microscopes. But basically what we see is that the transition of this occurs in a posterior to anterior wave. And it's fascinating to see how even the plasma membrane almost rolls over, and you can see it just in the bright field alone. So going forward, we wanted to do some work on what's the zinc signature status of the sperm cells after they populate the oviductal sperm reservoir. And so working with Dr. David Miller from the University of Illinois, we did some work with him to see if we included zinc in the media, can sperm actually be released? Is the release of zinc ions from the sperm cell necessary? And that's in fact what we saw. We did a series of experiments with less and less concentrations of zinc, and we saw that if there's zinc present at the concentrations of seminal plasma, we had no release from the oviductal sperm reservoir, and that we had to have lower concentrations of zinc that's present in the oviduct just for the sperm to actually be released and proceed forward with the pathway to fertilization success. And so this all came about because of Teresa Woodruff's group did a lot of work on the oocyte side. They showed that after fertilization, billions and billions of zinc ions are released from the oocyte. And we wanted to ask the story, where's the zinc actually coming from for the oocyte? And so this is some of the work that they did, and ultimately what they showed is that the amount of zinc that's being released from the oocyte corresponds with that oocyte's later ability to reach a blastocyst, or day 7 embryo state. And so that's kind of what inspired the work that we did. But it seems reasonable after we merge these two stories together that if capacitation is defined solely as the capacity to fertilize, it seems reasonable that the oocyte can decapacitate spermatozoa via zinc signaling pathway. And so this, in some respects, gets to a polyspermy defense mechanism. And so for decades we've known the slow block mechanism to blocking polyspermy, but we've not known the fast block in higher order mammals. And so this is important because if you have an oocyte that's fertilized with more than one sperm cell, then it's embryo lethal. It won't proceed forward to create an embryo and new life. And so what Teresa's group showed is after that zinc spark occurs, there's a 300% increase in zona pellucida zinc after that zinc spark event occurs. So there's a 300% increase in the amount of zinc on the outside of that oocyte. And so at the same time as we were doing these studies, others came out and showed that when sperm cells are attracted to the progesterone that would be present at the time of ovulation, that zinc can actually overpower that progesterone chemoattraction and turn the sperm cells around and go the opposite direction. And so it started supporting this theory that maybe zinc signaling from the oocyte can actually turn sperm cells away and be a quick polyspermy defense mechanism. And so we did a series of events to look at multiple enzymes that are present to make sure that the sperm cell can get through the egg or the oocyte. And we saw that one of those was MMP2. And when we did zymography experiments, we could actually see how zinc would block the activity of this enzyme that allows the sperm cell to make its way through the zona pellucida. Likewise with 26S proteasome, the 26S proteasome is another enzyme that allows a sperm cell to break down the outside shell of the egg. And we saw, as we increased the concentrations of zinc, that we saw less activity for the 26S proteasome to break down various components. So it all comes together to show that zinc regulates many important steps on the pathway to fertilization success. We have to have zinc present in order to have a sperm cell that has a high lifespan that's left. A zinc signature 1 sperm cell will have the most lifespan left, zinc signature 2 a little bit shorter after that, and so forth. But then also the ability to shed zinc reflects its ability to actually be attracted to an oocyte. And so this all came together to show that there's multiple sub-cohorts of sperm cells. And if we can analyze what sub-cohorts of sperm cells are present in a collection, that perhaps we can get a better indicator of fertility. And so in a really small fertility trial, we identified that higher fertility boars had a better ability to go through the zinc signatures after in vitro capacitation. So their ability to run the race, if you will, through in vitro capacitation, they had almost a twofold increase in the number of sperm cells after in vitro capacitation to reach a signature 3 state compared to the lower fertility boars. And so this was enough to warrant doing a larger field study. And so we worked with Acuity Genetics, and the data that I'm presenting here has 284 ejaculates across 81 boars, and almost 5,000 sows. The final data that we're doing the final analysis on is about 100 to 110 boars that we'll get through. So we ran those boar samples through in vitro capacitation. We looked at the 0 hour, the 1 hour, and 4 hours of in vitro capacitation, and then ran them through the image-based flow cytometer. And this allowed us to look at what percentage of sperm were able to run that race, what percentage of sperm cells can make the changes that are necessary to go through the events that mimic fertilization. And then we did machine learning and deep learning analysis on that to correlate it with fertility. And so this next slide here is just kind of an example of the image that we're able to get out of the image-based flow cytometer. We're able to look at what's the zinc status of the sperm cell. We're able to look at the acrosome the health or the modifications to the acrosome, look at the head shape with a DNA stain, look at the plasma membrane integrity as well as the live dead state, and then ultimately the morphology from the side scatter and bright field as well. And so ultimately what allows us, by looking at the multiple time points, we can actually subtract each of the subpopulations from the 0 hour time point and reveal what amount of sperm are able to go through fertilization-like events. And this really came out to show that every boar has differential ability to go through this final maturation event. And so here we see in green and yellow added together is the total number of sperm that were percentage-wise that were able to go through this maturation event. And we have some boars that have quite a bit of their sperm reaching that event in the 1 hour time point, and that's represented here in yellow. And then there's some boars that take a little bit longer to go through in vitro capacitation. And so it kind of gives us an indicator that maybe there's some boars that maybe they go through capacitation too quickly when they're in the sow. And then there's other boars that do a better job of living the race long and have sperm cells that might be present all across the entire ovulation window. And so this comes together to show that there's boars that have high capacitation ability, there's boars that have moderate capacitation ability, and then boars that have low capacitation ability, as well as a time component in there of quick versus slow. And so we were able to tell about almost 80% accuracy of high versus low boars when we did this study between the zinc signatures and the delta populations and the change in the populations through the zinc signature as well as the other assays we were looking at. And this was quite a bit better than just looking at motility alone. And so then finally getting into pipetting robots. And so before the pandemic, getting a pipetting robot cost about $65,000 starting out. And then right before the pandemic happened, Opentrons came out with what they called an open source pipetting platform. And so this is a $5,000 pipette that allows us to do quite a bit more economical pipetting. And then if you buy them used on the secondhand market, oftentimes from COVID diagnostic labs that are going out of business, you get them for anywhere from $500 to $800. And this can really streamline our diagnostic testing and make sure that we have uniform pipetting ability across a high number of samples. And so in the not so distant future, if we think about 10 years ago or even 5 years ago, when we posted an image on Facebook, it would ask us, is this John Doe? Is this Jane Doe? Now it doesn't even ask us if who the person is who it thinks it is. It just knows because it's so well trained. But we don't have that for sperm cells. And so if we're able to take the same techniques that they use for Facebook image facial recognition and use those machine learning and deep learning algorithms to sperm cells, it'll allow us to look at multiple biomarkers all in a matter of about 30 seconds going forward. And so just as a proof of principle, we did that with morphology, and then I'll show you some of the data that we did with biomarkers as well. But we know that there's quite a variability in the morphologies that can occur in sperm cells. And so we know that the consequences of abnormal sperm morphology are detrimental to fertility. And so we have to know the morphology of the sperm cells to have a baseline understanding of what's the potential fertility that's present. And this is just an example of the images that we use in the training model for the morphology differences. And so on a high resolution screen, we get quite a bit better resolution than the projector. But you can see examples of the distal mid-piece reflex, the distal cytoplasmic droplet, what a normal cell would look like going through the system, as well as proximal cytoplasmic droplets. And so working through the data analysis pipeline, we used Python as well as a package within Python called Keras that allows us to do deep learning, artificial intelligence research on that. That's all been packaged together today with Amnis AI that's a little bit more graphical user interface easy for cell biologists such as us. And what we were able to see is that as we moved up from the 20x, 40x, and 60x objectives, we were able to achieve over 99% accuracy rates of the different morphologies. So it provided a proof of principle that can we train a computer system to look at the morphology differences for what we wanted to do longer term. And so longer term, where we want to go with this is taking what they've done in the cancer cell world. And so cancer cell research is really moving towards a label-free diagnostic system. That way you can look at many, many more samples in a day without having to use expensive reagents. And so ultimately, there's small little characteristics of a cell that might be hard for the human eye to pick up on, but various scientific equipment can pick up on these small differences. And so what we do is we use a biomarker to train it from the traditional standpoint that this is a bad cell, this is a good cell, and then look at other features that we don't have to use those biomarkers, and ultimately take those biomarkers away so we don't have to spend hours in the lab before we can get a result, that we can get a result quite quickly. And so this is an example of various different biomarkers that we could be training the system on this for sperm cells. And we can see here with bright field contrast methods, various different bright field contrast methods allow us to see different features of the sperm cell. And so by incorporating these various different contrast methods, we might be able to pick up certain attributes of the sperm cell that another contrast method won't be able to pick up on. And so some of the preliminary data we ran through, we looked at the acrosome integrity. And so it's really hard to tell the differences between this acrosome and this acrosome in the bright field. But as soon as you put the biomarker on there for the acrosome, we can see that there is, in fact, differences between these sperm cells. This would be an infertile sperm cell as opposed to a fertile sperm cell. And we're able to train diagnostic models with a label-free approach so that we can look at the cell health within the matter of 30 to 60 seconds with 98.8% accuracy rates. And so the ability to do this in the future on a traditional microscope is quite promising, so that we can tell at the boar stud level what's the potential fertility of the boar as it comes in. And so some of the main takeaways, motility and morphology are bare minimum for analysis needs. We, by all means, need to look at motility and morphology, make sure that the cells can show up to the site, the job site. But biomarkers can tell a more accurate level of detail after we have the motility and morphology present. And then biomarkers are hard to implement in production, but label-free methods in the future might be able to help increase our ability to make this have stakeholder impact. And automated quality control sample preparation with pipetting robots is here. And so I'd like to thank our funding sources with USDA, as well as everyone in the lab, the grad students, undergrads, as well as collaborators. And with that, in the pursuit of life, liberty, and baked seeds, right here from Ames, Iowa, I'd like to thank you and take any questions.
I have a question.
Yeah.
So under the microscope, you're looking at a subsample of the larger sample, correct?
Mm-hmm.
And then from there, you're deeming them as infertile fed. Is there a way to remove the infertile or the ones that are marked by biomarkers from the entire sample?
Yeah, so the cattle world is using that more than the pig world. And so there's small magnetic nanoparticles that we can actually use to tag the bad ones and then pull them away with magnet. That's happening a little bit on the AI side in cattle, but more so on the IVF side, and especially as a cleanup method for like sex sorting sperm cells for X or Y chromosome. Yeah, good question. Yeah.
Carl, when you look at the bright field contrast methods and things like that, how many cells do you have to look at to say this ejaculate of how many billion cells is?
Yeah.
How many cells do you have to evaluate, given the fact that you have all these different populations and the variation?
For sure. So in the training models, to train the model at first, it takes, we've seen that after we get past 50,000 cells, anything more isn't needed. But then after that model's been trained really well, you can be down to about minimum of 300 cells, but more between 300 and 800 would be more favorable. Yeah. 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 Cool.
So this is something that would take computer automated.
Yeah. Hopefully in the future we foresee it as like a plug-in to a CASA system. Yeah. You have your motility output, your morphology output, and then your label-free biomarker. And then what's better as compared to the image-based flow cytometry, you'll be able to tell that on every single cell instead of just having averages across and adding them together. That way you can tell even if it has good motility but bad biomarker status, you can tell quite more defined. Yeah. Yeah.
The pool of bores that you used to do that big quantification study, was it a mix of terminal and maternal bores or specific lines or?
Yep, so that was through Acuity's test herd that they did. And so it's through the terminal side, so the Duroc line, and then minimum of 50 sows bred in fixed time AI situation. Yeah. Yep. So, cool.
All right, so let's give Dr. Kearns another round of applause.